Welcome, everybody. My name is Deepak Srivastava, and I'm happy to welcome both the folks here in person in the Meili Auditorium, as well as all our guests who are live streaming and are online. This is a very special day for us and for the international stem cell community, because this Ogawa-Yamanaka Prize that you'll hear more about has emerged over the last decade as among the most important stem cell prizes given across the world. At Gladstone here, it's one of our flagship scientific events, and you'll hear a bit more from Shinya Yamanaka in a moment about how this award came about, but it was through the generosity of Hiro and Betty Ogawa, who you'll hear more about in a moment. But they set up a $150,000 cash prize to the winner, and Shinya and I and our team here have crafted a beautiful memento that I'll say more about later that we'll be giving to our recipient this year as well. In 2019, we created a partnership for this award with Cell Press, and they've been wonderful partners since that time in helping us spread the word about this award and seeking nominations and then publicizing this. And there will be an article in Cell Stem Cell about this year's recipient. I also want to thank our selection committee, which is an international one, and we received over a dozen nominations this year, and after a lot of deliberation, the selection committee chose our recipient, and that committee is made up of George Daly, who's the Dean of Harvard Medical School, Hideyuki Okano, who's been the Dean at Keio Medical School in Tokyo, Lawrence Studer from Sloan Kettering, Fiona Watt from London, and Shinya Yamanaka and myself. And one of the privileges of chairing that selection committee is getting to call the recipient and give them the news. And Rusty Gage, who's our recipient this year, who I'll say more about later, is a close friend for many years, and it was a particularly special phone call to make this year. At this time, I'd like to welcome Shinya Yamanaka to the stage to say a few words about Hiro and Betty Ogawa, who were almost like parent figures, actually, to Shinya, and he'll say a bit about that. And just by way of background, for those of you particularly in our online audience, Dr. Yamanaka trained here at the Gladstone Institutes in the 1990s, made a seminal discovery of induced pluripotent stem cells while at Kyoto University, and for which he received the Nobel Prize in 2012. And we're fortunate to recruit him back to Gladstone to have a laboratory here since 2007. And he's continued since for the last 17 years as a senior investigator at Gladstone, and also for many years founded and led an institute in Kyoto called SIRA, which he remains director emeritus. So Shinya, please.
Good morning. So let me talk about Hiro and Betty Ogawa. They are our very good friends and also great mentors to me, to my wife Chika, and to all of my family. Hiro, he grew up in Japan. He got his undergraduate training from Kyoto University. But after that, he came to the States. He got his graduate training at Washington University, University of Washington, where he met Betty Ogawa. They fell in love immediately, and they got married. When Hiro was 49 years old, he started his own company about international ship container business, and he made his company very successful. Actually, he became the first Japanese who made his company public in the New York Stock Exchange market. He was very successful. So after that, Hiro and Betty decided to give back to society. They helped many activities including stem cells research, Gladstone, and also ISSCL, International Society for Stem Cell Research, in which Deepak, I, and also Rusty did presidency in the past. In April, 10 years ago, 2014, Betty and Hiro were in Japan, and after enjoying cherry blossom, they came to my office in Kyoto. And they proposed this prize. They say, Shinya, we wanted to start a scientific prize to recognize scientists contributing to stem cell research. And also, they made it very clear they want to recognize not only scientists, but also their spouses, because it is the spouse of the scientist who made it possible, who supported the scientist for the entire life. So that was their strong wishes. They had a name also. They proposed, Shinya, it's going to be Betty and Hiromitsu Ogawa, Chika and Shinya Yamanaka Stem Cell Prize. So I had to discuss a little bit about the name. Well, Hiro, first of all, I think it's too long. And more importantly, I believe you should have just your name, Ogawa, in the prize. But he insisted. So we don't have enough time because they had to get on the plane. So I told him, Hiro, I will be back in San Francisco in about 4 weeks. So let's discuss it again in San Francisco. But as soon as they came back to San Francisco, Betty was diagnosed as as the very late stage 4 cancer of the esophagus. And actually, like 2 hours before I landed at San Francisco airport, Betty passed away. So it turned out this award, and also the name of this award, to be the very last wish, one of the very last wishes of Betty. So we decided to have this name, Ogawa-Yamanaka Award. So now I really appreciate this name because every time we have this ceremony, Chika and I think about Betty and Hiro. So I'm so happy about this award. So this year, Rusty and Marilyn, you have met Betty and Hiro, so they must be very, very happy about this year's awardee. So congratulations and thank you very much.
Thank you, Shinya. At this time, it's my honor to introduce our 2024 recipient for the Ogawa-Yamanaka Stem Cell Prize, Rusty Gage. By way of background, Rusty actually had a very interesting childhood where he grew up in Europe and went to a boarding school there. And it was there that he had the good fortune of meeting his high school sweetheart, Mary Lynn, and they ultimately got married and have been together and just celebrated their 51st anniversary. And Rusty went on to get a bachelor's degree at University of Florida and then a PhD degree at Johns Hopkins, and then ultimately spent 5 very, very productive years at the University of Lund in Sweden, and made some of his initial discoveries around understanding the hippocampus at that time. Before he was recruited to San Diego, initially at UC San Diego, where he spent nearly a decade before moving over to the Salk Institute in the mid-90s, where he's been ever since, including serving as president the last 5 years at a key time in Salk's history. Now, over that time, Rusty continued to make really landmark scientific discoveries for which he's being recognized today. And while there are many, many different areas he's impacted, what particularly stood out to the committee were his contributions on recognizing early on that, in fact, in the human brain, there is ongoing neurogenesis, even in the adult, which previously was thought to not occur. And following that, after Shinya's discovery of reprogramming adult cells to stem cells, Rusty made the important observation that one could reprogram adult cells directly into what he called induced neurons, and that those could, and Marius Wernick, who's here in the audience and is a previous recipient, had made that initial discovery, and Rusty showed that those induced neurons could, in fact, retain an age clock in them that was erased when cells went all the way back to pluripotency and then were turned into neurons. And that was important because so many diseases of the brain are diseases also associated with the aging. And so having neurons in the dish that were aged, age appropriate, was key to modeling, and I think Rusty will say a few words about that. And more recently, he has really made inroads on understanding three-dimensional neurons in the form of organoids, which he'll speak about as well. He's published nearly 1,000 papers over his career. That's a lot of papers to write, and but he wasn't alone in that, I learned. And Hiro and Betty, as Shinya mentioned, always recognize that any success, male or female, is really dependent upon having a good partner across life. And it turns out that while Mary Lynn has been a wonderful partner at many levels with Rusty, she's also been a key contributor because she, in her own right, has had a professional career as an editor, graduating valedictorian from her high school class, I understand, and going on to be an important journal editor. And it turns out that every paper any of you might have read from Rusty's lab, or a grant that you might have reviewed, went through Mary Lynn first. And I'm sure that had a significant impact for all of us because it probably made it clear for all of us to understand. So thank you, Mary Lynn. Now, Rusty has, in addition to his scientific contributions, he's also played an important leadership role, not just as president of Salk Institute, but also as president of the Society for Neuroscience, many years ago, and as Shinya mentioned, president of the International Society for Stem Cell Research, through which he got to know Hiro and Betty personally. He's also served on the Gladstone Scientific Advisory Board for many, many years, and I know Leonard's group has benefited from Rusty's wisdom for many years and continue to as well. So Rusty's clearly had a remarkable career. But I want you to get a sense of Rusty the person as well, not just the scientist. And like Shinya, Rusty has been a marathon runner. He's been a long-time athlete. But what I just learned today, which I still find hard to picture, and maybe you have videos, but Rusty was a professional kickboxer till the age of 26. That's how he put himself through all his schooling and training. But you know, I say that because for all of you, for the folks that are young in the audience, you know, life is multifaceted, and it's important to devote yourself as you do to science and the things we do professionally, but it's also important to nurture all the other aspects of your life. And Rusty's also, and Marilyn, are proud parents of 2 children, Alexandra and Francesca, and I hope you guys are watching online as your lab and many other important people who've been important across your career. So please join me in welcoming our 2024 recipient of the Ogawa-Yamanaka Prize, Rusty Gage. We're gonna take a picture here, and I've got the memento for you and your money. Most importantly, one year I had to say the check was in the mail. So this memento, which I wanted to take a moment to describe, Rusty, was carefully crafted and thought about, so that those of you who are in the stem cell field will recognize the top of this, which is a Waddington landscape, where the notion in our field is that a cell might start at the top and as a pluripotent cell, and adopts a different fate as it rolls down a hill. And once it's in one of these valleys, we used to think that it could never cross over a valley and become something else, or go back up the hill and become pluripotent again. And Shinya's discovery really showed that, in fact, with the right cues, we could take a cell down in one of these valleys and move it back up the hill and become reset to a pluripotent cell. And my lab and Marius's lab showed, and Doug Melton showed, that in fact you could even cross the hill and go to a different valley directly without even going up the hill. And so we thought this was particularly valuable to the field of reprogramming. Nice. And so we made this with an artist, local artist here in San Francisco, specially for this award. So terrific. Congratulations. All right. Thanks very much.
Good.
But I will leave your check here.
Well, make sure I don't leave it here, right? Well, thank you. I have a few people to thank to start off with, and first of all, I want to thank Betty and Hiro for thinking about this prize altogether and making it happen. And I look at the past recipients, and I'm very proud to be part of those that have received this award in the past. See a couple of them here, Marius. Also want to thank Shinya for his discoveries. I mean, he started this revolution of reprogramming that continues in unexpected directions all the time. So I think it's not over yet. There are some other things coming along that are going to make it even more inspiring. And I think part of the issue is once you overcome this resistance to change, you begin to open your mind a bit and think about all the possibilities that are really there. And we thank Shinya for that. Thank the Gladstone, Deepak, and the rest of the team for putting this outstanding process together. And it's so accommodating and so warm feeling to be a part of this that I really do very much appreciate that. The committee, of course, got to thank the committee for spending the time to go through all the other fully qualified people and come up with me. Hooray, good for the committee. Getting prizes is wonderful, and I'm happy to acknowledge this, but the realities of it are that I'm a representative of people that have come through my lab, fellows and graduate students that really are deserving of this prize as part of it, and hopefully some of them are online listening, and I want to thank them for their contributions to what's been a great fun career for me. I also have been very fortunate to have really top-notch collaborators and mentors, like you heard Anders Björklund, who were early in my life important for setting an example for how you can be an outstanding scientist, but also a good person, and that this is really an important part of being successful in the real sense of being successful, is trying to help others move their careers along as well. As Deepak nicely said, secret sauce oftentimes is your family. My wife, Mary Lynn, has been a partner in many ways, and my two daughters, Francesca, Cheka, and Alex. It's a great team, and we have great fun together, and their support and just love has really made this ride enjoyable. So I'm very happy about that. I'm going to jump into some science and tell you some science, but I was suggested that I give you a little bit of a sharper history of where things came from, and that way it'll give me an opportunity to acknowledge a couple other people that I didn't acknowledge. So as an undergraduate, I started into my freshman year in a research lab serendipitously with Bob Isaacson, and my first introduction to hippocampus, and I was hooked. I was doing electrophysiological recordings in epileptic bunnies, and from there I moved on to his graduate student, Dave Holton, who was at Hopkins, so I had a lineage there. And as Deepak said, I was fortunate to get a Fulbright fellowship to work with Anders Björklund in Sweden, along with Ulf Stenevie and many other people that were emerging as stars at that time. Now, I went there to work with him on hippocampal plasticity, and we, early on in my career there, we discovered that there was actually nerve growth factor-like activity in the hippocampus, and we did this using transplantation techniques that he had developed with Ulf and others in Lund, and they were angling towards clinical application of transplants, fetal tissue for Parkinson's disease mostly. But after 5 years, I moved to UCSD, and this was an interesting time. It was 85, so molecular biology was just emerging for neuroscience. It hit other fields, but it hadn't really captured neuroscience yet, and UCSD was a real hotbed for that, and I managed to go back and take courses and sort of tool myself up for the next generation of techniques that were coming along. I learned how to genetically engineer cells and use trophic factors to do things. And that was the same time that we were introduced to this concept of adult neurogenesis and developed a bunch of tools because we were not really convinced it was happening. And so we developed some tools that allowed us to quantify this and measure it in ways that we hadn't done before. And then a series of wonderful discoveries that led to understanding how it's regulated, that these cells are really stem cells, in fact, authentic stem cells in the human brain. It's regulated by activity and by enrichment. Then when we showed this occurred in humans, I began to think about humans as a model system. And this was right around the time where iPS, not even quite iPS cells, this is just embryonic stem cells were emerging as a tool to study human biology and human genetics. We were really beginning to think it was really important that we look at the human genome when we're doing this. And then Shinya comes along and turns the whole thing upside down by allowing us to take from individuals their own cells and convert them into stem cells and then differentiate them into all kinds of stuff, which was catalytic for my career in terms of just the things that I was able to, diseases I was able to be attracted to, and I was collaborating with a lot of people at that time. In recent years, after being president, having to narrow my focus when I was a president at Salk, I began to focus on aging, and maybe because I'm getting older, partly, but also just this unmet need. So that's what I'm going to tell you a little bit about, is a journey of aging and my interest in aging. So here's a little outline. I'm going to talk a little bit about these induced neurons as a model of aging that Deepak talked about, and that Maria Ferenig early on had shown that this was a possibility. And then I'm going to talk about little surprising changes that occur with aging. Now that we can look at aging, find out the metabolism and cellular identity are really a feature of AD over and above aging itself, and discovering that AD really is a biological disease and not just a consequence of aging, and not everybody is going to get this horrible disease. Another surprise was that many of the changes that we see in Alzheimer's disease early on reflect cancer. It looks like cancer, and yet in one case you're going off to a proliferative state, whereas in AD it goes off into a senescent state. And it's understanding that transition that's really captured our attention at this point. And then learning about senescence has opened up a whole world for me, and it was pretty controversial as you were here originally, because we always thought that senescence had something to do with the decrease in proliferation, but neurons don't divide. So where does that fit into the whole story of senescence? So hopefully I'll tell you that. And if I don't bore you too badly and have a little time, I will show you some efforts that we've made on using 3D. So most of what I'll tell you is monolayers, but showing you how we're putting things together in a 3D setting to look at immunology. So aging is a major risk factor for many diseases. And if you go to an aging meeting, what you'll see is they're siloed off into people that think, well, no, it's all about telomeres. No, nope. It's about, proteostasis. No, you're completely wrong. It's epigenetic. No, it's energetics, mitochondria. And so we've had, fortunately, a group the last 9 years, 10 faculty members at the Salk, along with collaborators, who have a large grant to look at the interface between these different areas and try to look at the blend. And this has allowed for not just the multidisciplinary work on a system, but also bringing in some of our computational expertise to try to figure out how do you integrate quantitative data generate from completely different systems and make some sense out of it. So for Alzheimer's disease, the dominant theme for many years has been this tangles and plaques that were originally discovered by Alzheimer's. And the models that people use are either cells or flies or worms or mice that have been genetically engineered to express the human form of the disease in this genetic basis, or what we call the familial forms, that generate these tau tangles and A beta plaques. Those are really accounting for the familial forms, which is about 3% of the total population of individuals that have it. So 90% or more are what we call sporadic. So they don't have mutations in these diseases. They show some of the phenotypes, but there aren't any good models for what we call spontaneous, non-familial forms of the disease. So obviously, when we first started, we took a large group of humans aged from 1 to 90, and first to get an idea of modeling with these different age groups, made fibroblasts, made iPS cells, of which Shinya showed us we can make anything we want with these iPS cells, including neurons. And so when we looked at this, we saw in the fibroblasts we could find a lot of age-related genes, so things are uniquely expressed or misexpressed, I'd say, in people over 40 or over 60. The iPS cells, however, erased this difference in age, and we could not find differences also in the neurons. So we used a cocktail that originally Marius had, and then we made some additional changes in the media and timing of this, and developed a protocol for us to differentiate these human fibroblasts into neurons in 21 days. And this is sort of a fibroblast cluster here that you can see. And here we'll see, let's see. It was supposed to be done a little bit better. Just want to show you, see if you can see this, that you all would approve. There we go. So these is a time-lapse image of the cells. So what we do is we stably transfect the fibroblasts with a doxycycline control of NGN and ASCII2. So we can grow the fibroblasts to confluence and then turn on doxycycline, and you can watch them in the dish, and they just convert from a fibroblast into a neuron. It's pretty remarkable. So this is time-lapse over 21 days, so we're obviously condensing it down. So they go from this rather striking flat pace to neurons, and if you stain them for TUJ1, they're all neurons. So it's a true transformation. And when we do this, we see that there actually are a lot of cells now in this IN program that are differentially expressed in aging. One of the ones that we found with Martin Hetzer was this nuclear protein called RAMBP17, which controls the amount of proteins that can go inside and outside of the nucleus. And it declines when we get old in normal, healthy individuals, so that our nuclear membrane becomes leaky. And we know that this is an epigenetic phenomenon, because if you take old cells, 90-year-old people that have a leaky membrane, and convert them into iPS cells, you can repair that loss. So we have a small group of individuals working on molecules that can upregulate this leaky membrane, nuclear membrane. We've made these vectors systems available to a lot of people who've used them over the years, looking at a variety of things: epigenetics, stress, heterochromatin, proteostasis. We see that they really are there, and this is something that Deepak alluded to. Using this Horvath clock, you can see that the iPS cells are all day 0, whereas the INs are same age as the fibroblast, and they're comparable age to the chronological age of the individuals. We can do this through a variety of techniques showing that they are actually retaining the age of the individual. Another nice feature of this is genes that we care about in aging, like MAP, like tau, for example, which in its infancy, as a young form, is missing a couple of the exons. It's important with the INs, you capture those, so it's the mature form of the genes that are there too. When we looked at Alzheimer's disease, we had a large cohort of 19 non-demented and 16 demented individuals. We converted all of them and found that they all made a lot of neurons and inhibitory, excitatory neurons, small fraction of GABAergic neurons. And there really was no difference in terms of the efficiency of differentiation. The efficiency came, we started looking at the details of the genetic analysis. So we're looking at differentially expressed genes now using RNA-seq. No differences between the fibroblasts. Remember, these are old fibroblasts, and AD is same age, and there's really no difference at the fibroblastic level. However, when we look at controls versus AD in the neurons, the eye neurons, there's massive numbers of genes. So we looked at using a variety of tools. We see, for example, with simple principal components analysis, you can separate out the controls from AD. You see a couple of controls over here. It turns out that these guys had converted from after they had a biopsy and diagnosed, they became AD. So we're actually predicting ahead of time some of the phenotypes that are there. They have a very high correlation with existing RNA-seq data from postmortem tissue. There's lots of correlation between the genes that we find in INs and many of these other studies had shown. Even using single-cell sequencing, where we're looking at neurons primarily, and we look here at the neurons, and we see a very high correlation between their postmortem neurons and the genes that we find in our sample, even though they're different people, obviously. So what are the genes that are happening? So the first surprise for us was the genes that are downregulated. Maybe not so surprising. We see that there are identity genes. The neurons begin to lose, in the AD group compared to controls, they lose their identity. Things like synaptic LTP, synaptic transmission, plasticity are all downregulated in AD relative to controls. We can confirm this now that we have a cellular model and do synaptic measurements of postsynaptic, presynaptic densities are decreased in AD. Morphology has changed, and even the calcium imaging decrease. We've explored this a little bit more detail using single-cell sequencing or using patch clamping, and we can see that these are the young, these are the aged, which are significantly decreased, and the AD are even lower with regards to the action potential frequency and the actual amplitude. When we do evoked responses, so that was spontaneous activity, we get the same sort of changes that are reliably different between AD controls and aged individuals. So what's upregulated? Here's where our second surprise came, and that we look at the genes that are, this is just the differentially Go terms, the gene set analysis that we used. So Notch, HIF1 alpha, apoptosis, cell proliferation, NF-kappa B, so oncogene signatures. What is going on with AD when we see all this cancer-related phenomena occurring? We think what's happening is the cells are de-differentiating, and this means that they are becoming more, they're losing their fate, as we saw with their identity, but they're also de-differentiating into more of a stem cell-like character, if you can think of it that way. This group should think about it that way. So cell fate stability and epigenetic erosion is one of the concepts that's emerged for us. So doing a technique that allows you to look at the epigenetic landscape, we can look at ATAC-seq peaks, and we can see that in the AD they're significantly more open, and we can confirm that these peaks are open. So the chromatin is opening up, and the cells are de-differentiating. We look at it a little bit more closely, and we see that the openings are occurring primarily in the promoter region. That's the area of the genome or an individual gene which controls the regulation of its expression. So these genes, these cancer genes that we're were silenced and these mature granule cells are for some reason opening up. And we can even identify the DNA binding domains and show that another interesting thing is happening. Things like LDHA and HK2, which are glycolytic. This is another form of energy utilization. You have oxidative phosphorylation for very efficient generation of ATP, which neurons use almost exclusively. But here you see them shifting. So the neurons themselves are becoming more glycolytic, and they're opening up these chromatin regions that are allowing for this glycolysis to occur. In addition to RUNX1, HIF1 alpha, and these are all cancer genes that are also opening up. So there's a whole program shift that's occurring in AD that is coordinated, we believe, into what one would generally call an integrated stress response. So how do we know that this is really an age-related phenomenon? So one of the things that we can do is we can take the fibroblasts that we started off with from all these AD and control patients. And what I showed you, I converted them all the way over here. Are you watching my arrow? Okay, yeah. Converting them into neurons. We can use this same construct, which is the doxycycline-regulated control of NGN2, ASCII2. And after we convert all of these patients into iPS cells, we can convert them directly into neurons, also called IN neurons confusingly, but these are from iPS and these are from fibroblasts. And then we can compare these converted INs to these converted INs. The only difference being that we've now erased the age component. When we do that, and we look at the iPS cells, control versus AD, we don't see a difference as expected. But also the iPSCs are also not significantly different between the controls and the AD. So now we have an IN can capture the cellular aging, and it's required to detect differences between Alzheimer's disease, patient-derived neurons, and control, age-matched control individuals. AD induces a loss of cell identity and a metabolic reprogramming. We've done a lot of, there's a large, wonderful data set called the RASMAP data, which is made available for everyone, where they do single-cell sequencing of postmortem tissue, they have bulk sequencing, and we can do gene set analysis on our data and their data, correlate that with cognitive decline, pool the data together, and ask if there's any common sets that are existing between them. And once again, oxidative phosphorylation is down in both groups. Carbon metabolism, metabolic pathways are all up. Pathways for cancer are increased. There's another way you can look at each of the gene sets and look for a common gene that's expressed in each of these gene sets in both groups. And what we come up with a gene called PKM, surprisingly. So PKM is pyruvate kinase, muscle. There's two different isoforms of this gene. And the one form, PKM1, which is normally used, is very important for generating TCA cycle, pyruvate into the TCA cycle to get phosphorylated oxidative phosphorylation. PKM2, however, is an alternative splicing form, which occurs in cancer. This is a really big, important gene in cancer. And this is upregulated, or its activity is upregulated in AD relative to controls. And we've confirmed this not just in the ROSMAP data, but also in our cells. And we see this lactate to pyruvate shift that occurs. And we do a sort of reality check by getting good antibodies to PKM2 and going back to our postmortem human tissue to see whether or not this differential expression exists there. And sure enough, we see an upregulation or an increase in staining of PKM2. At a cellular level, you can see that there's significantly more staining of PKM2 in AD cases relative to the controls. So what we want to see, in following this up in a variety of ways, what happens is that PKM2 gets phosphorylated, and that allows it to pass in through the nuclear pore membrane. It's a smaller molecule. It's only a dimer. It gets in there, and it attaches to the DNA and stabilizes a bunch of really important proteins, including HIF1 alpha and STAT3, which are drivers, we think of them in cancer, but they're really drivers of glycosylation. So here we have a mechanism for why there is this shift that's occurring from PKM1 to PKM2. It's in part driven by this desire. I'm just safe for the side for aficionados to tell you one thing. We have a theory that we're working on for why this happens. There's a lot of DNA damage that's occurring in these cells, but I'm not going to be able to go into that story too long. But the nucleotide distribution is separate. So there's a disbalance between RNA nucleotides and deoxynucleotides. The only way you can get new nucleotides is to go back to glycolysis, go into the phosphate shunt, and pull out where you can generate biogenesis for lipids, amino acids, and nucleotides. So we think that this is an adaptation. The shift to glycolysis is an adaptation, just the way cancer cells do it. Cancer cells need that glycolysis to get all that protein and lipid to make more cells. And I think that this is a feature that's also true for cancer. So we're exploring this linkage between cancer and AD in a concerted effort at this point. I'm running a little bit longer than I should. We have searched in the cancer literature. They've discovered a natural product, a Chinese natural product called shikonin, which is available. It's also in Vietnam. You can extract this molecule, and it has this unique property of binding to PKM2, and forces a dimerization of PKM2. So it makes it into a tetramer, just like you see in a normal PKM1 version. When you do that, it can't get into the nucleus. It can't activate this glycolytic pathway. So you can end up reversing this. We have an assay system where you can measure PKM2 movement into the nucleus, and our shikonin can block that movement into the nucleus. And we do metabolic profiling, and this dysregulation of metabolic profiling we see in AD is also reversed with shikonin. And we do a whole RNA-seq on these that we show all these changes that I showed you early on about identity changes. Many of those identity changes are also shifting in the IN cells compared to the controls. So we think we have a mechanism now for, we believe that PKM2 is a good mechanism. I'm working with Lou Cantley, who is instrumental in identifying PKM1-2 shift in cancer, and we're looking at different ways in which we can tease out these things for developing therapeutic interventions. Another observation was made by Joe Hurdy in the lab, and he was looking at these Go terms in AD and noticed that like 25% of them had to do with senescence. And what does senescence have to do with any of this? So he took the gene expression patterns and went back to the senescent database and looked at the RNA-seq data, and many of these genes that were upregulated in AD are true senescent genes. And we look at that data enrichment score in the INs, they're all highly expressed, not in the IPS-INs and not in fibroblasts, but uniquely here in the INs. So giving us some more feeling that let's follow this. So Joe followed it further, and first to get a good validity check, he looked at P16, which is not going to go into it, but it's a good marker for senescence in the actual senescent field, proliferative senescent fields. And what he sees is a nice localization of new N-positive cells with P16. A bit of a surprise, because we thought that most of the senescence that would be going on there would happen in glial cells. We do see it in astrocytes, we do see it in microglia, but we think that's a consequence of the whole thing may be starting in neurons and cells. So we've identified a senescent-like event occurring in the human brain, and we wanted to see if we could exploit this in our model system. Do we see this? So another marker people use is beta-gal, and sure enough, we see an increase in beta-gal. And in vitro, we also, in our IN models, we see an increase in P16 and the AD relative age-matched controls. A whopping difference between AD INs and AD INs and IPS-derived INs. There's orders of magnitude more P16, and this is obviously true with beta-gal as well. So one of the features of Alzheimer's disease and senescence is the inflammatory response that occurs. And the thought is that this is a interferon gamma, NF-kappa B mediated event where the cells get activated and they start secreting cytokines. Those secreted cytokines then can affect the surrounding astrocytes and microglia and induce an inflammatory response in them. So we did find, in fact, there was this nice upregulation. We took conditioned media from our cells and also CSF fluid from AD patients and from patients with age-matched controls that Doug Glasgow had stored away in our ADRC. That's an Alzheimer's Disease Research Center that we are the stem cell core for. And sure enough, when you look at CSF and you look at the IN conditioned media, we have a high correlation between factors which are within this domain of what are considered to be inflammagens or inflammatory molecules. We can even take the conditioned media from our, so not only are they expressing the proteins, but is it biologically active? We take that conditioned media directly, we don't condense it or anything, put it onto healthy, mature astrocytes, and you see an activation of GFAP. And when you do RNA-seq, you see things that you're probably more familiar with IL-6 and other things are upregulated in these cells by virtue of just taking the conditioned media from the IN cells and putting on the cells. So again, moving quickly through this, there's a whole strategy nowadays of thinking about senolytics and xenomorphics. Senolytics is going to try to take a senescent cell and turn it back into a good, healthy cell and living the good life. That's xenomorphics, sorry. Senolytics are the ones that get rid of these cells. So this is a strategy that people have begun with, and in fact, there are 2 clinical trials that have been published using a particular senolytic cocktail in Alzheimer's disease patients. The problem is that they didn't check blood-brain barrier, and neither compound got across very effectively, so it didn't have any effect, but it didn't hurt them, which we are most afraid of when you're going in there and killing cells. But we did take this combo of quercetin and desmoid and gave it to our cells, and sure enough, we were able to show a dose-dependent increase in tunnel expression in the ADs relative to the controls. I'm not sure this is a strategy I would go for, but it does highlight the fact that there are these senescent cells, neurons, that may be initiating, and these are all neurons in this case, that are initiating a spreading effect. When we go back into the brains of these patients, we can see little splotches in the postmortem tissue of senescent cells, and it's as if a group of cells become activated, and then they start affecting the cells around them. We've treated our glial cells, for example, with conditioned media multiple times, and they turn into P16 positive too. So that inflammatory experience can induce a senescence type response in the glial cells. So one way to think about it that we're challenging ourselves with is could these shifts that we're seeing, an increase in damage, the increase in of inflammatory response, be sort of the initiating event that leads to some of the phenotypes that we see with AD. And so our view of this right now is that, just to give you a bottom line and where my thinking is, we actually think, you always wonder, where does this whole thing start? And in another set of experiments, I would tell you and make the case it's really genomic instability, which is the beginning of this, that leads to the epigenetic erosion, senescence, inflammation, stem cell exhaustion that will be the future of where we're going. Do I have a couple minutes left? Okay. So I'd like to share with you sort of a parallel line of thinking, and that is all of what I've told you about so far is monolayers. And we're learning a lot, and we're mixing other cells into our monolayers now. We're even making three-dimensional INs packed with astrocytes and microglia as a way of enriching them. But we've gone back to our organoid work, or back to a three-dimensional traditional iPS-derived organoids, with the goal of trying to make it more like a human brain, so all the constituents that we would like to have. One of the issues that became prominent for us when we started doing organoids about 10 years ago was the core. If the organoid gets larger than about 2.5 millimeters, there's a diffusion gradient, and nutriments can't get into the core, so the core begins to rot. And that rot carries out, but many people that are looking at organoids 4 months, 5 months, they're really only assessing the rim around the outside of it, not in the core itself. This bothered me. I went back to my old way of thinking in transplantation as many people have taken these to chips to try to get them vascularized through engineering approaches. I did it old school, which is to try to get them transplanted into a non-skid mouse into a site that we discovered back in this paper in 1984, where it was very richly vascularized. So if the superior colliculus is in the ventricle and it's highly vascularized, and Ulf and I found that when you place tissue into it, in those days, fetal tissue or superior cervical ganglion, they would get vascularized and survive very well. So we did this with our transplants. We'd make a transplant in vitro and then transplant it into this core, and you can see here that we can inject a dye into the interorbital space, and the blood flows through very nicely and without any leakage with this dye. So we suggested that not only did they survive very well in the core, and there was no cell death, but also they were vascularized and a blood-brain barrier was formed. We looked inside more recently and have developed a technique for putting microglia inside the organoid. So we make the organoid, we take primitive microglia in the erythroid progenitor state. They migrate into the graft, and then we graft it. And you see these beautiful microglia that are sort of patterned throughout the human organoid, really large and quite robust. And they take on a lot of the properties of a true microglia that I've been working with Chris Glass and his team for some time, trying to characterize the specific nature of microglia, human microglia, in an in vitro setting. And here we get to skip over that and put them into a human setting, and so they display many of the properties of a human microglia. Working with Axel Nimmerham, who had discovered 10 years ago that microglia are not static cells. They're called surveillance cells. And when they're in a static state, they survey the environment by moving their little tentacles around, testing the body. And they never make contact with any of the other tentacles. They just survey the whole environment. So we make a glass cover slip over the top of the mouse and he builds these tiny 2-photon microscopes that you can attach to the head. So the animal can be walking around with this microscope in his head. And this is the sort of thing that you see, that they're live cells that are just surveying their environment. You can see that they're not touching each other, just making so there's something to be learned about how they touch each other. But anyway, but they are reactive. If you give LPS, they turn from these really nice surveying cells to this macrophage-like cells. So Axel has this other thing that he does, where he'll survey the environment, and he takes a laser and he kills one cell in the middle between them. And what mouse macrophages or microglia do is they move in from a distal location to the damaged area to gobble it up. But this is what human cells do. So here they're sensing their environment. He kills the cell, and they send their processes in, and leaving their body in the same location so they can continue surveying on all parts, but they send part of their processes in to kill it. This is a dramatic effect. It's quite reliable, and we're pursuing this in a variety of other things. But I'm going to skip this for time and just say our goal really is to try to make these cells, make these organoids a little bit more authentic, as authentic as we can possibly make them. So we've developed a way to imbue these organoids with human astrocytes as well. If you don't do that, mouse astrocytes will come in. But if you induce the cells to make their own human astrocytes, it prevents the mouse cells from coming in. But what is not prevented is the endothelial cells and parasites. So our blood vessel that we make inside this human brain is human astrocytes, but mouse endothelial cells and mouse parasites. And here's an example of just what that looks like. When you do 3D reconstruction of the EM, this cell is the astrocyte, this is the blood vessel, the endothelial cells, the parasites, the basement membrane, and the astrocyte, which completely ensheaths it. This is also different than mouse, where the astrocyte forming the blood-brain barrier, 2 or 3 astrocytes usually clip it because they're about half the size as a mouse. But the human astrocyte can completely ensheath this. So we're still, this is the end. This is not the end. So we have to get our next step, which is to get the human blood vessel organoids. So we've developed and confirmed a technique that other people have shown, which is what's called a blood vessel organoid, where you can make from iPS cells, you can make endothelial cells, purified endothelial cells and parasites. We then sort them out from each other and reconstitute them back into the appropriate percentage or ratio that you see from human brain, which is 3:1 astrocytes to parasites, whereas in the periphery, it's 1:11. That's why you have no blood-brain barrier in the periphery. So we can isolate them, and they have all the right properties. And we are now building our models. I left this slide out. Building these models to show that we can get these human endothelial cells to migrate in the anastomose with the mouse blood system and can supply through these vessels blood into the organism. We're almost there. We're doing the best we can to get that done. Just to think of the next steps, we've got to humanize completely the vascular system. We need to make oligodendrocytes. We need to get these guys myelinated, and then we can, I think, more completely model a disease when we have all the cell constituents that we need. Thank you for celebrating this with me. I hope I give you a little bit of a flavor of the kind of work that we're doing and hope to be doing for a long time coming. Some of the key people is Mayan and Lei, who really drove the astrocyte work. We have a fantastic course, like I'm sure you have here, that are just indispensable for many aspects of the work we do. And Simon Abed for the original work on forming the organoids in vitro, and for Simon building the microglia system into quite a workable neuroimmune system. And with that, and your kind attention, I'm done.
Thank you so much, Rusty. It was wonderful. We do have time for some questions from the audience. Steve? We'll use the microphone so the online audience can hear.
Great talk, Rusty. Thank you so much. I had a question. It's sort of two related questions. It had to do with the cell identity work that you did, because that's been reported in other diseases too, Huntington's disease and things like that. And so in the back of my mind, I wondered whether this is a response to neurodegenerative disease. And sort of related to that, there's also been some suggestion that, you know, the effect or the effect of aging conferring risk of neurodegenerative disease is well described. But some people have even suggested that neurodegenerative disease may accelerate aging. And so kind of that sort of idea, I just wondered what you thought about neurodegenerative disease evoking some of these things, or how that all works together.
Yeah. So I'll restate what I sort of alluded to. It's a crucial question, and it's always, who's the starting point? And so one way to answer the question is that there's a homeostatic mechanism of all these different hallmarks of aging, and that any perturbation in one of those systems can have an impact on another, and they funnel down into a common thing. I'm okay with that. It's taking it back with your socks on. But I'm more thinking about our data really supports the fact that there's really a DNA damaging issue, and that this initiates in almost every cell that you see an integrated stress response, which one of the things that happens is the ribosomal biogenesis starts decreasing. If you look at your data, you'll see ribosomal biogenesis is almost one of the first things to shut down. It takes up 70% of the ATP in the body to make proteins. And so they put all the machinery into compartments, holding off their activity until stress goes away. But the point being that there's also a lack of DNA, there's lack of nucleotides that are being made, especially in neurons, I think, are particularly vulnerable to this because they don't have cell division, so they can't make deoxynucleotides that way. And in AD, we published on this last year, there's a very important system called ribonuclease reductase. And that's the system that takes RNA, ribonucleotides, and converts them into DNA. I mean, it seems like a simple process of just taking off the hydroxyl group, but it's complex. There's 4 of them, and it has to be done in the right way. That's down by 70% in AD. That enzyme complex of 6 enzymes is down in AD. So not only can they not get nucleotides from cell division, they can't salvage RNA into DNA. So I think this is, for me, a driver right now, because the only other way they can do it is to go back into glycolysis. They go into glycolysis, they go to phosphate shunt, they generate nucleotides that way. And that's what this one of the things that we feel is really important. Your point of, it's going to be a cascade. So when that sort of stuff happens, you're going to get a lot of other things happening. And there's going to be vulnerability in, I do think of amyloid deposition and tau deposition are responses to these kinds of stresses that are going on in the cell. Particular cells that are particularly vulnerable to that in disease states. But I'm completely biased, I admit it. I'll go with the neutral view, which is it's all one thing or another, and they all come down to a common form. Or I'm pushing this idea of vulnerability of DNA. And sort of think of DNA stability as sort of the, in Star Trek terms, the prime directive of a cell is to protect your DNA. And so everything else is sort of revolves around that attempt to save your DNA from being damaged.
Makes sense.
Long answer.
Sorry. Questions? Yes, Mara?
That way.
Thanks for the talk. It was really enlightening, really interesting. I have a question about the same type of structure, the PKM story.
Yeah.
So one of the it actually acts as an enzyme, right? So a metabolic shift is actually a common trend for most cardiovascular and neuro diseases as well. And was interesting to me, if I understood correctly, actually changes from an enzymatic activity to an isoform that becomes transcriptional regulator.
Right.
So my question for you, how common that is? And are we missing by actually thinking as metabolic and transcriptional as kind of independent fields, right? So we think about things that will change metabolism by changing metabolites that actually affect
right.
the phenotype, and then not about transcriptional changes that you demonstrated.
No, you're absolutely right. And I have to admit completely that when we came across this shift that occurs in this imbalance that occurs in PKM, I had no idea what this is about. I mean, I always thought PKM1 was this enzyme that's important for oxidative phosphorylation. But you're absolutely right. PKM2 is a smaller molecule. And it's not so much a transcription factor as it binds DNA and stabilizes DNA for other transcription factors to do it. I mean, it's a nuance there. I think we're missing a lot. I think if you look in it'd be interesting to look into Huntington data and see whether or not you see any kind of metabolic shift that's going on that might involve this as well. I think it's a broader attempt on the part of a cell. I'm going back to this integrated stress response because there's so many features of that are sort of evolutionarily conserved for the cell to emit when it's undergoing stress. And I think at the beginnings of all these diseases, we see it. Then you might have a mutation that makes you vulnerable to go down one direction, or you might have a life experience or something taking down one direction. I may be oversimplifying, but it's helping me to follow my thoughts right now and keep my thoughts together. So I think you're absolutely right. I think that we've missed that point. We sort of think of the transcriptional areas. That being said, there are 3 transcription factors that are massively upregulated in AD that are underrepresented. RUNX1, YY1, which I'm very excited about, and PDRQ is up. And we're exploring, could they be, what's driving them, and what's so I think there's lots of really cell biology that needs to be done here. I could say more, but I'm not going to.
Maybe I'll ask the last question. And Rusty, along those lines, there are obviously byproducts of metabolism that affect epigenetic regulation, like alpha-ketoglutarate. Do you think that there may be a connection with that?
Yep. Well, yes. So one of the things that I didn't talk very much about, I alluded to it, is this concept of epigenetic erosion. What that really means is that there are large complexes that keep things packed. And I think that some of these metabolites, these energy metabolites, are also influencing that. One of the set that are most important that I'm excited about are line elements, which we've worked on for many years. They play a role in chromatin silencing, but then they're silenced by factors that are released or eroded, and they come up. So when they're lifted, then you start seeing a lot of activity occurring around areas where line 1 was acting as a suppressor. So, and then of course, once you have line 1 being expressed or being made available, that might be also contributing to the senescent-like response, because it's a foreign. If it's translated, it's foreign. And even double-stranded version of it is going to be attacked by the CS thing.
Great. Well, thank you. Well, these kind of events take a lot of effort by many, and I want to take a moment to thank Irma Hernandez and our communications team for putting this all together. And again, thank the Ogawa family, including Andrew and Marcus Ogawa, who are their sons, who are now supporting this award. And finally, please join me in congratulating our recipient this year, Rusty Gage.